Okay, so these graphs are rheological data. So basically they show the storage and loss modulus, or the elastic and viscous component of shear modulus of the material.
And it's on a logarithmic scale?
Yeah. So what we're looking for is that the overall modulus, the bolded line, is at the right sort of grain level, and that is just above this line. So it being above this line makes it a gel that makes it sort of able to hold its shape. It being, the main line being at this point makes it about brain tissue-y.
Okay.
And it being sort of consistent means that it's not, if you had lines that sort of crossed over, that's when you get into those viscoelastic, non-Newtonian type fluids, and you wouldn't want that to happen, because then you start dancing, and then all of a sudden your brain would be like solid. What's going on? Why did I inject myself with cornstarch? That would be weird.
Why would you inject yourself with cornstarch?
I don't know.
It gives you a weird high, apparently.
Especially when dancing.
Apparently, that's what someone tells me.
I know, I think it makes your brain a
Ask
bit
a friend.
I don't know.
Injection.
Maybe you're just like really keen on supporting Darwin effect. Take one for the team, as it is. Okay, so now putting it all together, this again will look a little bit similar to what we've seen before, but now we have two different growth factors. They're similar. One is GDNF. It's basically just a different one so that I can detect the difference between the two. It's glial cell line derived neurotrophic factor. Again, very good for up and coming new brain cells. Says be a brain cell. Okay. But the point is that this GDNF was unmodified and the BDNF was modified, and you can see again that you have the BDNF, or GDNF, sorry, the unmodified one coming out initially, the BDNF modified one coming out in a delay. And what's cool about this is they were both in the same material, so once again we need to be paying attention to make sure that the individual system we're looking at will work with the whole system we've already seen and established. So in this case, we definitely need to make sure that those chitosan chains we've attached to BDNF won't hug all of the other proteins, or won't, mess up the gel such that everything will delay it, or be delayed from releasing out of the gel. So here, with the modified and unmodified drug being in the same hydrogel to begin with, and then both showing the release profile that we wanted them to show, we know that we can independently control the drugs. So it's not just that we can decide to put this drug in and control when it will come out. We can put multiple drugs in and control when each one will come out, regardless of the other ones, which is cool.
Is this from in vivo studies?
No, all of this is not even in vitro at this stage. It's all ex vivo. It's basically, it looks really exciting here with the plots and stuff, but what it looks like when I'm actually doing it is a little plastic plate full of a whole bunch of holes, and I put some gel in it and some water essentially on top. And every hour, because you notice these 12-hour hourly things, so it takes about 4 hours to set this up, and then me coming in taking like 10 samples every hour on the hour for 12 hours.
That sounds tiring.
Yeah! It's fun. Weird stuff goes on campus late at night.
At least it's not a 48-hour experiment.
Yeah. Okay, so that is actually all there is. This is the rest of my crazy lab group, and they're wonderful, and thanks to them, Dr. Dave Nisbet, Dr. Claire Parrish, Dr. Richard Williams, funding, and the labs. Woo!
Yay!
Yeah. Any questions?
Will you be moving on to in vivo studies? Or?
Yes, I will, actually. We're currently plotting out our in vitro stuff, which will happen within the next sort of month, probably, to make sure, basically with the in vitro, what we want to do is make sure that the modification doesn't remove the bioactivity of the drugs. So we have a hint at that because the method we use to detect them is called ELISA. It's enzyme-linked immunosorbent assay, and it uses antibodies to detect the protein. So the fact that they're still recognizable by antibodies suggests that they're still in that right functioning shape, but it's not absolute proof. So first we're going to test it with cells, make sure that the cells, like just in petri dishes, when introduced to the modified, unmodified stuff that's been through the gel, they respond as if they're still receiving regular old BDNF, which they should. And then once we have that demonstrated, basically the clinching final bit of my PhD will be demonstrating that this does work in vitro, or in vivo, sorry. So that'll be the just before I finish.
What's in vivo?
Will you be doing it in mice or?
Yeah, it'll be in mice. In vivo means inside a living organism, and then in vitro is just with living cells that aren't the whole organism, they're just the cells.
Cool.
So would you intentionally damage a rat's brain and then implant the...
Yes. So how it works is you get, well, mice more than rats actually.
Yeah,
But yeah, you, depending on, my stuff is nice and broad in its application. It's just for general, like whenever you need to deliver drugs to a particular area of the brain. So I'm focusing on stroke at the moment just because that's a relatively easy model. So it's sort of like damage in this region. But you could also be using it for certain neurodegenerative diseases such as Parkinson's, affects dopaminergic neurons specifically. They're located at like a specific region, so you can sort of use this drug delivery method there because it's very location specific. But that's harder to replicate, it's very specific. With the stroke, it's easier, so that's the main reason I'm focusing on stroke. But yes, you get a mouse and you do give it a brain injury. You confirm that it's had that injury, so you do sort of like behavioral assessment on it and make sure that it's, you know, walking with one side of its body and stuff. Yeah, there's no real nice way of explaining this, but that's the way it works. And then after you've confirmed that you have injured them, then you try to heal them. Well, some of them are control groups of various stages. And then you again see if that's worked. And even if they have worked, you still have to kill them to look inside their brains and see how it's integrated on a cellular level.
Can we get the brains to synthesize the molecule on its own?
Sorry, what was that?
Can we get the brain can we get the organism to self-synthesize the molecule needed for repair?
No. So there are certain things. Oh, like the BDNF, you mean?
I mean the
Or
whole thing.
this,
Just insert a gene that encodes for a similar protein.
So you can do that for the drug delivery portion. You do that by viral vector delivery. The problem with that is that it's permanent. So if you want something going on,
can't you put a trigger in front of it that says stress triggered? So under stress, certain different transcription factors will be blah blah blah.
you might be able to. That goes outside my scope of genetic engineering. But the main point of this project is that it's going to apply when a structural support material is already needed. So while those things might be possible, we're going on incorporating with this material that's definitely needed.
Could that be used outside of the brain? For instance, like spinal injuries with the nerve damages to spinal cords and whatnot?
It can actually, it doesn't have to be limited to nervous system tissue at all. Any sort of tissue engineering material can have this sort of drug delivery system incorporated into it. You lose the sort of, it's really vital with this system because of the blood-brain barrier, but it's still useful in other systems as well to be able to control the temporally different drugs coming out of the material.
Would it work with eyes? Because they're the other thing that's quite isolated from the blood.
Testicles as well.
Yes.
In theory, this is all, again, outside my scope. This is where I play the engineer, not a biologist. Don't actually know any different systems. But anywhere that you would use a tissue engineering material, specifically hydrogels, which is pretty much everywhere, then this could be applied.
Would you need to take into account normal fibrosis formation? Like in normal brain repair, you'll get fibrosis after a certain injury, certain conditions. Would that factor into this at all?
Yes, likely. We're at the borderline of my biological understanding. Are we getting into sort of scar formation here with this?
No, essentially.
Yeah, so you would need to take that into account. You'd hope that this would be applied sort of immediately after, or else this would be applied, either you'd have a different strategy for dealing with the scar, or you'd may have to just sort of remove the scar and then that would become your lesion site and then you put this in. But mainly we're focusing on using this before a scar has a chance to form.
So
this
more
is immediately.
trauma than neurodegenerative.
I focus on them because they're simpler.
Cool. Thanks for the speech. It was awesome.
Thank you.